So Dembski, having said that, you might have thought he would be dying to show up in court and play a role in the squeezing. Well, there's the cover page of the lawsuit, and when the lawsuit was filed, 8 expert witnesses lined up for the other side in the trial. Here they were. Now, when you volunteer to be an expert witness in a federal trial, it's not a trivial procedure. You have to write an expert statement. My expert statement was 75 pages in length. It was about the length of a master's thesis. It took me 3 weeks to write it. Dembski did the same thing, and all of these guys had to do that. And then you get to be deposed by the other side, which has that expert statement in hand so they can see what you're going to testify and ask you about it and so forth. And I told you my deposition lasted 9.5 hours. Same with the other 5 expert witnesses on our side of the case. Then what happened is the lawyer showed up at the place arranged for William Dembski to be deposed, and they walked into the room, and Dembski wasn't there. And a guy walked in with a briefcase, said, "I'm Bill Dembski's lawyer. Dr. Dembski has withdrawn from the case." Then they went to Seattle, Washington, the guy who was fantasizing about the vice strategy. They decided they would depose Stephen Meyer, the director of the Institute for the Center for Science and Culture at the Discovery Institute. The lawyer said, "Sorry, Dr. Meyer has withdrawn from the case." And one at a time, almost all the people who were affiliated with the Pro-Intelligent Design Discovery Institute who had committed to defend the school board pulled out of the case. They ran. They didn't show up in trial. These 3 people did, and I give each of them a lot of credit for having the courage to fulfill the promise that they made to the Dover Board of Education. But I still wonder why the leading lights of intelligent design, like Stephen Meyer and Bill Dembski, after promising they would show up, simply didn't show up at trial. It made the trial very interesting. The result, in any respect, is the trial demonstrated the complete collapse of intelligent design as anything even remotely resembling a scientific theory. That collapse was evident in the judge's decision, which came at the end of the trial, and that decision, which is called Kitzmiller versus Dover, is a stinging rebuke for intelligent design. And I would urge all of you to get a copy and read it. All you have to do is type Kitzmiller, K-I-T-Z Miller, into Google. It's the first document that comes up. And I would urge you to read it. It's 137 pages long, but it's double-spaced, so don't be put away by that. It's written in such plain English, you don't have to be a lawyer to understand it. It's very straightforward, and parts of it are simply laugh-out-loud funny. And in fact, it's so funny that several people who read the judge's decision and parts of the trial transcript actually emailed me and said, "Man, they ought to make a movie out of this." Well, the answer, folks, is 2 movies. Paramount Pictures has a screenplay under development for a movie based on this trial. I don't know how soon that will come out, but Nova has actually filmed a 2-hour show on this, which will air this fall, which includes a courtroom docudrama reenactment of key points in the trial. So if you don't want to read the book, you can wait for the movie, and the movie's got to be out really very shortly. Now, I want to show you the scientific basis of this argument from intelligent design and why it has failed to gain any adherence of note within the scientific community. Part of the claim is that evolution cannot explain the origin of complex cellular machines. Things like these beautiful and intricate flagella that certain bacteria like E. coli have. Now, why is it that they are able to make the claim that evolution can't explain it? Here's why. These structures are said to possess a quality called irreducible complexity. And irreducible complexity means they couldn't have been produced by evolution even in principle. Now, that's different from saying, quite correctly, that evolution has not yet explained, the evolutionary biologists have not explained where these things came from. That's true. But it's the claim that evolution will never be able to explain it because irreducibly complex structures can't evolve. And examples of such structures include the bacterial flagellum and the cascade of proteins that clots our blood every time we get a cut, a hemorrhage, or a bruise. Now, I want to go a little bit further into this. What does irreducible complexity mean, and why couldn't irreducibly complex structures have evolved? Michael Behe, who's the champion of this argument, basically argues that an irreducibly complex structure is one in which it's composed of multiple well-matched parts, and the removal of even one part effectively causes the system to stop functioning. It no longer works. So as he explains it, the reason that's an argument against evolution is because you just can't make an irreducibly complex system like this one. by numerous successive slight modifications of a precursor system. In other words, you can't take bits and pieces and evolve the complex system. Why? Because any precursor to an irreducibly complex system that is missing a part, even one part, is by definition non-functional. There are about 40 proteins in that flagellum. You can't evolve 35 and have a partially working flagellum, 36 a little bit, so forth and so on. You got to put all 40 parts together or you get no flagellar activity. He argues that couldn't have evolved, it must have been produced by a designer. It's a powerful argument. The problem with it is it's wrong, and it's wrong in a most spectacular way. And the example I want to give you tonight, because of limited time, is this complex of proteins that clots the vertebrate blood. I don't know how many people in this room have ever taken a graduate level course in biochemistry, but the pathway that you see here is one of the mechanisms by which biochemistry professors torture their students. I was made to memorize this pathway when I was in grad school, and I've hated that professor ever since. But it's an intricate system, and it's really quite beautiful. Now, here's what Behe says about this. He says none of these proteins, there's almost 20 of them, all these proteins have to be necessary. None of them are used for anything except controlling the formation of a clot. These fibers are part of a blood clot over here. In the absence of any of the components, blood does not clot and the system fails. That's a very powerful statement. Now, is it just sort of a stray statement? No, it's at the heart of the argument from intelligent design. This is Pandas in People. This is the textbook that was given to the Dover students to study. There's that blood clotting pathway. Same statement. Only when all the components of the system are present and in good working order, all the components, does the system work. Some of these proteins, we know how these things evolve. These are all, almost all of them belong to a class of proteins known as serine proteases that were probably formed by gene duplication. Does that mean they derive from one another? This is that textbook. It might be. But all the proteins have to be present simultaneously for the blood clotting system to function. That is a scientific claim. All the proteins have to be there, and it's testable. And you know what? We can test it in nature. Here is a detailed diagram that I made to make it a little bit clearer of all the proteins in the blood clotting cascade. The boxes are the ones that are serine proteases. These are the proteins that are so similar that they were probably made by gene duplication. But let's do an experiment, and let's eliminate just one of those factors. Remember the claim. They've all got to be present, or blood doesn't clot. So I'm going to propose we eliminate factor 12. I drew the box around it. Well, guess what? Nature has already done that experiment for us. Because it turns out there's a class of organisms that doesn't have factor 12. Those organisms are whales and dolphins. They lack factor 12. And guess what? Their blood clots perfectly. So that claim that they all have to be present is just wrong. But you might, well, it's just one. You know, do you have anything better than that? Well, the answer is we certainly do. Let's try it again. Except this time, let's take away not one, not two, but 3 parts from the system. And you can see the 3 parts right there. And once again, nature did the experiment for us. It turns out that the pufferfish, known as fugu, is actually missing 3 of those parts. And guess what? Their blood clots perfectly well. So the claim that all of these things have to be together or blood does not clot is wrong. Now, this in itself doesn't show how the evolutionary pathway came about. So if you wanted to say we're not sure about evolution of this pathway, I'd say, right, mazel tov, you got it. As it turns out, we are sure for a different reason. But that claim that they all have to be present in the absence of any of the components, blood does not clot and the system fails, that claim is simply wrong. And that's one of the things that came up very quickly in trial. And this is one of the reasons why this claim has not gone very well in the scientific community. It also turns out that the very same study out of the laboratory of Russell Doolittle in San Diego actually did show how all of these components were formed by gene duplication and by a mechanism that geneticists call exon shuffling. And the interesting thing about it is Doolittle's lab also worked on an organism called the sea squirt, which is very closely related to the ancestors of us vertebrates. Sea squirts don't clot their blood. They don't have a vertebrate clotting system. But it turns out that nearly all of the parts of these proteins, which today clot in vertebrates, actually were present in the sea squirt. Meaning the parts were already there. They were performing other functions in this organism. And what evolution did was to assemble them into a working clotting mechanism. In other words, we really do understand how this mechanism came about.